in protein expression comes out drastically as a difference in systematic level. With this, we are going to start the session. Yes, sir. We can start.
Yeah, thank you very much. Hiten, can you please stop sharing session, your screen, so that I can start sharing my screen? Is it visible? Yeah.
Yes, sir. It is visible.
So, as Kaushik introduced, I am going to talk about proteins. I would like to go back to the basics of the biology. We have learned how the cell divides, how many organs develop, and we have also learned about plants. But all these things are taken care by some basic molecules in biology. We know these basic molecules. We know about DNA, we know about RNA, we know about proteins. So in the next 15-20 minutes, I will try to discuss how proteins are very important for every different kind of biology what we have discussed today. For example, right now I am talking to you guys, right? So I am talking, you are understanding what I am speaking, and as soon as you are understanding that, some questions are, you are having some questions in your mind, and all these things are happening. These are basically some signaling events are going on. In our brain, in the surface of the neurons, there are some protein complexes. We call them ion channels. These ion channels changes their structure and accordingly allow ions to go inside another neuron, or some neurons releases some ions. And due to these kind of events are happening, some electric signal is generated that is again received by another protein in the cell, in a different cell, and that is reacting. So all these things, whatever happening here, are nothing but some jugglery of some proteins. Okay, so now what are proteins? So proteins, we can tell proteins are some large biomolecules, large polymers. We know some unnatural polymers which are very much popular nowadays and creating lots of pollution. Those are plastic polymers. But nature designed these proteins as the mostly used polymer molecule. So proteins are naturally selected polymers. DNA, RNA, these are also natural polymer. Proteins are another kind of naturally selected polymer. Okay, so and how proteins are basically important. So proteins, when these polymers can take different kind of structures. I am going to discuss these different kind of structures in the next 15 minutes. So the structures are known as primary structure, secondary structure, tertiary structure, quaternary structure. So the same protein molecule may have different kind of structures in order to function. Okay, and the same protein molecule, by the virtue of having different structures, may also do different functions. So the structure of the protein is also very important. Proteins are everywhere, as I have mentioned. So proteins is life. Protein is life. I mean, even if we are not going for bodylifting, our body is composed of proteins everywhere, from hair to nails, from blood. Hemoglobin is one of the most popular protein molecules. All of us know about it. It carries oxygen. In the brain and nerve cells, there are proteins. All the enzymes, those are proteins. Antibodies. I mean, against COVID-19, we have to develop some kind of vaccine. So to develop these vaccines, we are basically, when we are injecting this virus in our body, or dead virus or attenuated virus injected in our body, some different kind of proteins are basically being produced in our body, which is going to give us immunity. Those are antibodies. We have cellular messengers, we have muscles, all the muscles are definitely protein. So all the bodybuilders eat lots of protein. Okay, so coming about structural proteins, you can think of mustache, you can think of hair, you can think of spider web, and you can think of this nice feather from a peacock. Yeah, so you can see different colors in this feather, and all these different kind of colors are coming from different kind of proteins. They are pigmented, and that is why we are seeing these colors. So again, different proteins, different structures, different color, different function. In terms of functional protein, there are many functional proteins. I have already discussed some, but But I just want to point out one protein here, which is known as Rubisco. Rubisco is the largest abundant protein molecule on the earth. It is present in the leaves, and what it does, the successful folding of Rubisco fixes carbon dioxide, okay, fixes oxygen. So that is the most important activity of the plants, what they are doing for us. In addition to that, we know that we have insulins. Many diabetic patients take insulins, and this is another interesting protein. This protein is known as green fluorescent protein. It was initially isolated from this kind of jellyfish, and this protein has a unique structure so that it fluoresces green. Okay, and what scientists have done currently, they have taken this protein out from this particular organism. How to take this out? Basically, we know the gene of this particular protein. We have cloned this gene in various other systems, and at the end of my talk, I will show how GFP is used in different systems to visualize different kind of things. So, as I mentioned, proteins are polymers, and these polymers are made of something called amino acids. Amino acids are chemical moieties which has a central carbon atom. Then we have an amino group, and the other side we have a carboxylic group. So one side of the carbon atom we have amino group, in the other side we have carboxylic group. And the last one, so we have a R group. We call this a side chain. This is the variable group. In all the amino acids, whatever we find in nature, this particular part is constant. Only this R part is the variable group. And depending on the R, the side chain, the amino acids have different kind of properties. We have nonpolar and hydrophobic amino acids. For example, here you see glycine, alanine, valine, leucine, isoleucine. These 5 amino acids are nonpolar amino acids. Then we have also hydrophobic amino acids, methionine, phenylalanine, tryptophan. And you can see a bulky side chain, aromatic side chains in these amino acids, which make them very hydrophobic in fact. And there is a unique amino acid called proline where side chain interacts with the amino group. Okay, all these different kind of amino acids mentioned here are mainly heating water. On the other hand, there are polar and charged amino acids. These are hydrophilic in nature. As you can see, there are acidic groups and the positively charged groups in these amino acids, which are basic groups. 3 basic amino acids and 2 acidic amino acids are present in the proteins. So altogether there are 20 natural amino acids. There are 2 more unnatural amino acids known, but mainly these 20 amino acids in various permutation and combinations are linearly arranged to make these protein polymers. So this organization of these amino acids in the proteins makes a protein that particular protein. So decides the identity of that particular protein. And these amino acids, when they come together, so how do they come together? They come together by a fantastic chemical bond, which we name as peptide bond. So what happens, let's say this is the first amino acid and this is the second amino acid. The carboxylic group of the first amino acids interact with the amino group of the second amino acid. It releases of water and this particular bond, peptide bond, is formed. Okay, and this is a fantastic strong covalent bond which linearly arrange the amino acids in their sequence. And once this sequence is done, then these amino acids in this sequence, this chain, they start interacting with each other, mainly via side chains, but also other interactions happen. The first kind of structure we see in these amino acids, okay, it makes this linearly arranged chain in helical structure. We call it alpha helix. Sometimes we see this kind of sheet-like structure, and this is named as beta sheet. So these are known as secondary structure. Please remember the kind of interaction involved here are mainly non-covalent interactions. Again, I remind you the linear change of amino acids are formed by covalent interactions, and here we have mainly non-covalent interactions. Now these secondary structures, they can further interact with each other. There is one covalent interaction which is important here, that is disulfide interaction, disulfide bond formation. But other than that, again, non-covalent interactions are preferred for this tertiary structure formation. Primary, secondary, and tertiary. So one linear chain can generate three different kind of structures: primary, secondary, and tertiary. And once this tertiary structure is formed, this tertiary structure can interact with another tertiary structure. So two units of protein molecules come together, or multiple units of protein molecules come together to form a quaternary structure. Again, this structure is mainly formed by non-covalent interactions. So we have the primary structure. We can call this kind of skeleton of this particular protein molecule. The primary structures interact via non-covalent interactions. The amino acids in the primary structure interact via non-covalent interactions and form the folded structure. And folded structures, when they are interacting with each other, then we have the functional protein, these happily dancing zebras. Now here, one thing I want to point out, if you see, other than the formation of the linear chain of amino acids, majorly non-covalent interactions are preferred in biology, are preferred by nature. Why? So, so formation of non-covalent covalent bond requires high energy expenditure. On the other hand, formation of non-covalent bond does not require that much of energy expenditure. So breaking this bond and forming the bond is easier than forming a covalent bond. So nature evolved in such a way it preferred non-covalent interactions so that the flexibility of these protein molecules are also maintained. The protein molecules remain flexible depending on the condition. They can change their structure or organization by modifying some non-covalent interactions and react to environmental stimulus. Okay, so these four different kind of structures basically determines protein's function. And if the structure is lost, this could be via change of a single amino acid. How a single amino acid may be changed? All these amino acids are basically encoded by our DNA. Okay, in the DNA there are particular codes which dictates which amino acids are going to be formed. So we call them genetic code. Okay, so if the genetic code is changed, the amino acid is going to be changed, and this change of genetic code, the process is known as mutation. For example, here I am showing a linear chain of amino acids, which is basically a lysozyme sequence. This is the amino group and this is the carboxylate group, and lysozyme is the enzyme which is present in our tears and mucus and helps to kill by different kind of bacteria. A single change in amino acid in this particular sequence will change the lysozyme structure, and lysozyme will lose its enzymatic function. Okay, and just one change. I can give you a famous example here. This is the example of sickle cell anemia. Here you see there are 7 amino acids. There are more amino acids in this hemoglobin chain, but I am showing here only 7 amino acids. So in this place there is valine, histidine, leucine, threonine, proline, glutamic acid. Now instead of this glutamic acid, somehow in a mutation this valine is introduced. As soon as this valine is introduced in this peptide chain, hemoglobin peptide chain, as you can see from this fantastic structure of hemoglobin which has become disorganized, and the sickle cell structure, a sickle-like structure of these red blood cells are being observed. These are red blood cells. The structure of the morphology of the red blood cell changes dramatically by the change of a single amino acid. Why this change? If you remember, I talked about the charge of the amino acid. Glutamic acid is a negatively charged amino acid, and here we have changed it with a neutral amino acid. As soon as it is changed to a nonpolar amino acid, it is trying to hide from water. Okay, when it is trying to hide from water, it changes the structure of the protein, which is forcing the RBCs to demorph and form this sickle shape. Similarly, a protein structure can be broken, devastated, disorganized by various different kind of stresses. Here I am giving a very simple example of egg. If you have an egg, just cut it open and start boiling it. After 1 minute, you can possibly see this kind of condition of these egg proteins. Egg white is mainly made of a protein called albumin. Egg yolk is also, contains also many different kind of proteins. So once you start heating this egg with different times, increasing time, you can see there will be change in the composition, in the texture of the and morphology of this egg proteins. You can see a very nicely boiled egg at this stage. So the protein content here to here are same. I am not ignoring there are other molecules like lipids and carbohydrates in egg, but protein is very high, and this is going, but the protein content remains same. But if you are heating this particular protein group of proteins here in the egg, it is changing its consistency, changing its texture, and becoming a very different kind of things. You can do this experiment with even omelette, even egg poach. In every cases, egg proteins are going to change in their texture in a different way, depending on the nature of stress you are giving to these proteins. So we call it, this kind of devastation of protein structure, we call it protein denaturation. Okay, many times due to different kind of stresses, protein denature even in our body. Okay, let us go back to the original knowledge what we have. We have in our cell DNA, as I said, the DNA codes for amino acids via RNA. This RNA is translated into different kind of amino acids. The process of DNA to RNA is known as transcription, and RNA to protein is called translation. And the translation happens on some molecular machines called ribosome. So this is the information and this is the final output. This is the information molecule and these are the proteins of the executor molecule, and this basically defines the information flow in the biological system. If there is a problem in the information, there will be change in the executor molecule, and once the executor molecule changes, they may lose its function, their own proper function. Going back to another aspect of biology, we have our body. I mean, our body is composed of around 40 trillion cells, 40 trillion different kind of cells. And if you think about the different kind of cell types, we have more than 200 different kind of cell types. If, as I have said, DNA, from DNA we have RNA, and from RNA we have protein. Please remember, in all these different kind of cells, the DNA content remains same. But what changes is the information flow. So number of proteins of the the different kind of proteins being expressed in different kind of cells. If you go back to a cell contains almost 70% of the water and 15% protein. If you see of other biomolecules, these biomolecules are comparatively much less in abundance than protein. So proteins are that important. Okay, and these proteins are coming from the information from the DNA. And the entire protein component of a cell, we call it as a proteome. Like possibly you have heard the word genome. Genome contains around 20,000 genes. Human genome contains around 20,000 genes. This genome translates for proteins, and a cell contains almost 10,000, 1 lakh to 10 lakh different kind of proteins. If you think about the number of proteins in a per cell, it may be in the order of 8,000 to 10,000. Here I am giving a very classical example. You can see a caterpillar and a butterfly. Both these stages of life contain the same genome, but the proteins which are expressed at this particular stage and the proteins which are expressed at this particular stage are entirely different. So the proteome changes, genome remains same. We have different kind of life stage. This is also true from tadpole to mature frog. In our body, as I had mentioned, we have different organs, and different organs have same genome but different proteome. We cannot allow the proteins designated to express in eye to express in liver cells, then it will be, a massacre in that sense. But there are definitely some proteins which are expressed throughout this body. We call them structural protein, okay, or homemaker proteins or housekeeping proteins. But most of the proteins are not housekeeping, and they are expressed in different organs, different kind of cells, and even as we have discussed today morning, cell division. Okay, at different stages of cell division, different kind of proteins are expressed, and the proteome of the cell is changing in order to show the proper morphology and function. Now, whether a protein can get sick? Yes, a protein can get sick. In fact, let's say, as I have exemplified protein as an active zebra, we can get a zebra sick, and once the zebra is sick, it can lose its color. So this we call a misfolded protein. and whether there is a system for this misfolded protein to be corrected in our cell. Do we have hospitals in a cell? And indeed we have hospitals in a cell, and there are doctors which are known as protein chaperones or chaperones. There are very different kind of chaperones. For example, in our society, we have different kind of doctors. We have gynecologists, we have child specialists, we have eye specialists, we have heart specialists. Similarly, in the protein society, there are different kind of chaperones which can take care of different kind of sicknesses of the proteins. Okay, and these doctors try to correct this protein, do surgeries, do whatever they want to do, and many times, or most of the times, I would say the protein comes back to its active and fully functional form. But in many cases, doctors fail. In our society, doctors fail, and in the protein society also doctors fail. And then these proteins are diverted, dead proteins are diverted to crematorium, and there are specific machineries inside the cell which work as crematorium for proteins. We call them proteases. There are molecular machines called proteasomes. There are particular systems called autophagy. These are different kind of crematoriums in the society, protein society. Depending on the budget you have, you can take your protein to any of the societies, any of the crematoriums. Okay, from less sophisticated to highly sophisticated crematorium for proteins. But this all societal problem, when we age, basically, all the societal problems are accumulating with time, and the protein society collapses. That leads to aging. That is one of the major reason of aging. And in case of some deadly diseases for aging population, most of us know that Alzheimer's disease, Parkinson's disease, there is another disease, another famous scientist, Stephen Hawking, who suffered from the disease, amyotrophic lateral sclerosis or ALS. We had ice bucket challenge few years back. There are Huntington's disease, Lewy body dementia, many different kind of age-related disease are accelerated because some proteins get sick. Okay, and for each of these diseases, one or two proteins become majorly sick. Okay, and then it imposes a problem in the cellular proteome society and kills the cell, kills the neurons mainly. There are diseases which also happens in muscles, but mainly neurons in this kind of protein sickness are showing their phenotypes. Okay, so in fact, if you see this with age, we are going to lose our muscles and everything. So the change in the cellular morphology again is dictated by the changes in the cellular proteome. And I want to end by showing one of the famous proteins which is responsible for Parkinson's disease. This particular protein gets sick in this disease. And you can see it, instead of a normal structure, I am sorry I forgot to put a normal structure of this protein here. This protein forms rods. And as we see this in the transmission electron microscopy, we tag this particular protein with the GFP molecule, as I have discussed earlier. GFP has particular structure which process and helps us to see these proteins under microscope. So when we tag these proteins, we find this fantastic mesh-like organization or woolen ball-like organization of this protein, synuclein protein in the cell. And this is not the correct form of this particular protein. It linearizes, it polymerizes, this protein polymerizes here and form this cane-like morphology, the woolen ball-like structures. Yeah, and the GFP protein is also used to visualize many other things. As you can see, we have stained these particular animal called C. elegans with GFP, Drosophila, and many different. This is zebrafish, mice. These are the individual cells in culture. So we can even see stem plants with these GFP proteins, and we can see in dark. Yeah, so proteins are mesmerizing molecules, and proteins are dictating our life. With this tone, I want to end. Thank you very much.
Thank you so much, sir. That was a provocating talk. So we have a question here. Santosh actually wants to know what is polar amino acids. So I would like to modify this question: how the amino acids can be differentiated into polar, non-polar amino acids. That will be interesting for the school students.
Yeah. As I mentioned, these proteins are composed of amino acids, and amino acids are mainly the backbone of the amino acids is constant throughout among these 20 amino acids. But only if there is something called side chain or R group of an amino acid, which changes from amino acid to amino acid. So depending on the molecules, atoms you have in the R chain, it can love the water or it can hate the water. So that determines the polarity of the particular amino acids, depending on the side chain chemistry over there. If I want to explain it better, then I possibly require a board here, which is difficult.
Yes, sir, I get it. So that was a very wonderful talk, sir. Especially, I love the way that you compared proteins with zebrafish and with some other examples. I hope that it will be a very interesting session for the school students too. Thank you so much, sir. With that, we are going to go for the next